Okay, thank you very much, everyone. So today we are going to talk about the writing and reading of the histone code during transcription initiation. We have previously discussed that when transcription needs to be initiated, the histone code is modified so that somewhere closer to the Tata box, so that other regulatory proteins, regulatory molecules, transcription, those that are related to transcription can bind to the Tata box and initiate the transcription. But for this, we need to relax the chromatin. So after this chromatin is relaxed, we need to change the histone codon for the initiation of the transcription. So how do these different proteins, they read or write the codon during this process? We have discussed that previously as well, that sometimes even the RNA polymerase carries some reader-writer complexes, and it keeps moving along the DNA and keeps changing and keeps modifying the histone code. So how does this happen? Let's assume that we need to initiate the transcription. So this is a histone, this is a nucleosome. So you see 4 histone proteins, H2A, H2B, H3, and H4. And these are their tails for each of them. So, so here we have 2 tails for H4 and 2 tails for H3. So this is H3. This is the second H3. This is one H4, and this is the other H4. We are not talking about H2A and H2B. So only about H3 and H4. So what I mean, here we have Tata box, and here we have the binding site for the regulatory protein. So what happens that a gene regulatory protein, it binds to its binding site upstream of the Tata box. So it is binding here. And then what does the gene activator protein do? It would recruit a histone acetyltransferase. We have previously discussed that these gene activator proteins, or these regulatory proteins, when they bind, they can recruit histone modifying enzymes. And these histone modifying enzymes can then later modify the histone code on the chromatin. So that's what we are talking about in details. Let's go back to this slide. So a regulatory protein or an activator protein binds to its regulatory sequence. Then it recruits histone acetyltransferase. So this is an acetyltransferase. So we know that this is going to acetylate some of the residues on the tail, some of the protein residues on the histone tails. So this is H3, and we have the second H3 on the other side. We have discussed this, that these histone proteins, this is an octamer, and it consists of pairs of these proteins. So H2A, H2B, H3, and H4, they exist in pairs. So pair means we have one H3 here, and we have a second H3 on the other side. So
excuse me sir.
Yes, please.
सर कुछ स्टूडेंट्स ज्वाइन नहीं कर पा रहे,
कुछ स्टूडेंट्स ज्वाइन नहीं कर पा रहे।
सर सुंदर ने मैसेज किया, वो काफी ट्राई कर रही है।
ओके जी, सुंदर एडमिट हो गई है। लेट आते हैं ना, लेट क्यों आते हैं भाई? तो क्लास चल रही होती है तो आई कैन नॉट लुक एट द पार्टिसिपेंट्स ना। सो एनीवेज, सुंदर को आई होप उसने ज्वाइन कर लिया। सीरियस स्टूडेंट्स है, क्या टाइम पे आ जाया करें।
सर मैं तो फौरन ही आपका मैसेज फॉरवर्ड कर देती हूँ।
सुंदर को 2 दफा एक्सेप्ट किया है, उसके नेट में इशू है, क्या प्रॉब्लम है? नहीं, आप लोगों को जो ही लिंक शेयर होता है तो ज्वाइन कर लेना चाहिए ना। ओके, सुंदर हैज जॉइंड। ओके, शुड वी प्रोसीड फर्दर?
यस सर,
चलें आगे चलें।
जी सर।
ओके, सो काइंडली आप लोग अपने माइक्स म्यूट रखा करें, कोई क्वेश्चन हो तो अनम्यूट कर लिया करें उसको। Clear to everyone? Okay. So a gene activator protein binds to its regulatory sequence, and then it recruits a histone acetyltransferase. We know that acetylation relaxes the chromatin, and it also facilitates transcription. So this lysine number 9 at 9th position, this on histone 3. So H3, K9 means H3 means that the this is the tail for histone 3, and K9 means lysine number 9, or the lysine, the ninth residue on the tail. So and K means it's lysine. So that would be acylated, and it would be acylated on both the histone tails. So we have a pair of these histone proteins. One H3 is on this side and one H3 is on the other side, and the DNA is wrapped around it. So both of them are acylated by this histone acyl transferase, and this histone acyl transferase also acylates the lysine, 8th lysine on H4 tails. So these are two tails of 2H for each, one on this side and one on the other side. So now we have got 4 tails acylated, 2 H3 tails and 2 H4 tails. On H3 tails, we have lysine at 9th position, that is acylated, and on histone tail number 4, on H4 tail, we have lysine on the 8th position, that is acylated now. So what happens next? This regulatory protein, this activator protein, now recruits a histone kinase, and we already know that kinases they phosphorylate the protein molecules. So kinases are the enzymes which have the ability to phosphorylate different protein molecules. So what happens now that this histone kinase is going to phosphorylate serine 10 on H3, so on both the sides. So on the 10th position, it is, so on the ninth amino acid was lysine, which was acylated, and next to lysine on position number 10, the 10th amino acid is serine. Now serine is phosphorylated. So what we need to keep in mind, what is important, is that this histone kinase cannot phosphorylate serine unless the lysine on 9th position is acylated. So this is important, that lysine on the 9th residue, the 9th amino acid, which is lysine, this must be acylated before you can phosphorylate S10. So S10 is phosphorylated on both the sides now. So this is a sequential matter. First, K9 or lysine on 9th position needs to be acylated, and then S10, serine 10, can be phosphorylated. Now, when both of these two things have taken place, we have phosphorylated on histone 3, we have acylated lysine on the 9th position, and we have phosphorylated serine on the 10th position. In addition, on histone 4 tails, we have acylated lysine on the 8th position. So three different types of modifications have been carried out. This sets up a histone code for transcription initiation. Now the code is ready now. On H3, we have, no, sorry, one additional thing we need to do is, after serine 10 has been phosphorylated, histone, this is histone acyl transferase. This would now acylate another amino acid, which is lysine on 14th position. This is now acylated. So now the codon is ready for gene expression. So this can be now read by the general transcription factors or other chromatin remodeling complexes. This is what we call that the histone code for transcription. This is the histone code for transcription initiation that you have on H3. You have now the K14 and H3. H3 is phosphorylated and K14 is acylated. If you switch back to, let's switch back to one of the previous slides. You see here, this is the code for gene expression. When we have serine at 10th position as 10 phosphorylated and K14 acylated, this was the code for gene expression. So that's the H3, that's the histone tail. And here we have all of them. So this is the K9, and this is S10 phosphorylated, and then this is K14. This can either get acylated or methylated. So this needs to be acylated. So when S10 is phosphorylated and K14 is acylated, that's a signal for transcription initiation that leads to gene expression. So let's see what's happening in our case now. It's exactly the same. We have S10 phosphorylated, and we have K14 acylated on H3 tail. So that sets up it sets up our histone initiation codon. When this is set up, now the transcription factor 2D, which is a general transcription factor, and a chromatin remodeling complex, what we call switch and SNF. So this is a complex of two things. One is switch and the other one is SNF. So this chromatin remodeling complex, what we call switch and SNF, this binds to the nucleosome, and transcription factor 2D binds to the Tata box. We know that this binds to the Tata box. So in order to bind to the Tata box, it's necessary that transcription factor 2D reads a histone code on H3 tail. And the histone code that transcription factor 2D reads is S10 phosphorylation and K14 acetylation. So unless this happens, we also have acetylation on K9. So 9th, 10th, and 14th amino acids, these three modifications are necessary on the histone 3 tail. So 2 are being acylated. Which ones? Lysine at 9th position and lysine at 14th position. These two need to be acylated, and serine at 10th position, this needs to be phosphorylated. So once this is achieved, this can be read by the transcription factor 2D. So this is a codon. This is a signal for the transcription factor 2D. Transcription factor 2D would read this signal on H3 tail. And this reads it by a domain, what we call a bromo domain. It is mentioned here. This domain that binds to the histone tail, the domain of the transcription factor 2D that binds to the histone tail, this is known as bromo domain. And this bromo domain is responsible for reading the histone codon or histone code. So unless this is achieved, transcription factor 2D cannot bind to it, cannot recognize its histone code, and hence it can also not bind to the Tata box. So this was a transcription initiation code for the binding of the transcription factor 2D. On the other hand, switch and SNF, they also bind to the other side of the histone proteins, and both of them together, now we have switch SNF, this chromatin remodeling complex, and we have got the transcription factor 2D. Then they trigger the rest of the transcription process, and you already know about it, that transcription factor 2A and 2B and 2H, they are recruited later. We have previously discussed this, but this is how a histone code is read and is written during transcription initiation. Did you follow everything, or do you have any questions, please?
Clear, sir.
Clear to everyone? Anyone else has any questions, please?
No, sir.
Okay, that's clear to everyone. Okay, thank you. So let's proceed to the next slide. I think this one was easy, not so difficult. I can also repeat it once again. So we have this Tata box here, and we have a activator protein binding site. So activator protein binds to this, it's a regulatory sequence, and it recruits histone acetyl transferase. Histone acetyl transferase would acetylate lysine on 9th position on both the tails of the histone 3, and on H4 it is going to acetylate the lysine number 8 or K8. Then this recruits histone kinase. Histone kinase then phosphorylates the serine on 10th position, but this is only possible when K9 is acylated. Afterwards, when this is phosphorylated, S10 is phosphorylated, then histone acetyl transferase can then can acetylate the K14 on both the sides of the H3 tails. After this is achieved, this is now the histone code for transcription initiation. Then transcription factor 2D and chromatin remodeling complex, which is known as switch and SNF, both of them bind to the nucleosome at two different binding sites. So The chromatin remodeling complex, it recognizes both the histone codes on H3 and H4 tails, while transcription factor 2D recognizes the histone code on H3 only. And both of them have got these binding sites, the sites that recognize these histone codes, and these binding domains or recognition domains, they are called as bromodomains. So transcription factor 2D or chromatin remodeling complex cannot bind to the nucleosome unless this codon is written, this code is written on the histone tails. And then transcription factor 2D can bind to its starter box and recruit the other general transcription factors, and then later RNA polymerase 2, so that transcription can be started. Okay, let's go to the next slide. So, excuse me. So here are 6 ways we have previously discussed, 4 different ways how gene activator proteins could, so here's the slide. These are 4 different ways how gene activator proteins could initiate transcription. They could carry out the nucleosome sliding, they could carry out histone removal, they could carry out histone replacement, or they could change the patterns of histone code. So these are the 4 different ways how these gene activator proteins could carry out the transcription initiation, and that's the 4th one we studied in details right now. Now we are moving towards the repressor proteins. What would they do? How would they modify the chromatin, and how can they express the activation of the proteins or expression of the proteins? So the first thing we study here, the first mechanism is competitive DNA binding. So here we have 2 binding sites. One is a binding site for the activator protein, and second is the binding site for the repressor protein, and both of them are upstream of the starter box. We have previously discussed this, that starter box is somewhere in the proximal region, or in the core. This is inside the core perimeter, and then we have proximal control elements, and then we have distal control elements. So these are the trans-regulatory sequences. So we have in this region of the DNA, we have a binding site for both of them, a binding site for an activator protein and a binding site for a repressor protein. But the interesting thing is that both of these 2 binding sites are overlapping. For example, the binding site for the activator protein starts from here and it goes all the way till the middle of the binding site for the repressor. So this is the sequence starting from here, going all the way till here, which can be recognized by the activator protein. And then the binding site for the repressor protein starts from here and goes a little into the, or inside the binding site for the activator protein. So both of them recognize the overlapping sequences. So here is a sequence for the activator, and here's a sequence for the repressor, and they're overlapping. So here is a here is the competition between the activator and the repressor. So that would depend upon the affinity of the binding proteins. Which one has a stronger affinity, whether repressor has a stronger affinity for the DNA, or it is the activator which has a stronger affinity for the binding of the DNA. So the one which has a stronger affinity is going to bind to the DNA more firmly as compared to the other one. Or sometimes it is like on the first come and first have basis. Whatever binds first doesn't let the other one bind to the DNA. So the repressor can either bind before the activator binds to it, or repressor has a stronger binding affinity, so it can remove the activator and bind to the to its regulatory sequence, and this is how it can block the activator from functioning, and this can repress the transcription of the gene. The second mechanism we have is masking the activation surface. So now here in this case, the binding domains or the binding sequences for both the proteins are distant from each other. They are distinct, they are not overlapping. But what is interesting here is that both activator and repressor are bound at the same time. But repressor has the ability that it can mask the activation surface of the activator protein. So both of these proteins have 2 different domains. One is a domain, we have previously discussed this, that the transcription factors, they have 2 domains. One is a DNA binding domain, and the second one is activation domain. So for both of them, for both the activator and the repressor, these 2 are the DNA binding domains, the domains that recognize a particular sequence on the DNA and helps them to bind with the DNA. And they have an activation domain. So this is an activator. It will bind to its binding site via its binding domain, and it has got an activation domain, which helps in activation or in recruitment of other proteins. Like we have discussed in the previous example, that this regulatory proteins recognizes its regulatory sequence on the DNA, and then this is the activation domain of the protein, and activation domain helped in recruitment of the histone acetyltransferase. And in another case, it also helped in the recruitment of histone kinase. So this activation domain can now be blocked by the repressor. Now, here we have got a repressor which is bound in the neighborhood or next to the activator protein. Now, this repressor has the ability to mask the activation domain or block the activation surface of the activator protein. This is how it can suppress the expression of the genes. The third phenomenon we have is the direct interaction with the general transcription factors. We have previously discussed that we also have some regulatory sequences which are lying distal from the promoter region, and for example, enhancer sequences. So what happens with these enhancer sequences, that an activator protein binds to them, and then this is the binding site of the activator domain, and the DNA can loop back bending proteins. Some bending proteins can bind here. We can switch back to one of our previous slides. We have already studied this. Let's have a look at it. Yep, here. So we have studied this, that sometimes these are these enhancer regions, they are lying farther from the promoter region, and they have got these binding sites for the activators. So activators bind to these enhancer regions. So this is the distal control element, and this is the proximal control element. So this is the promoter region. So because it is lying far away from the promoter region, the DNA needs to loop back onto the promoter region, and this is achieved by the binding of the DNA bending proteins. And then activators can, when bound to the enhanced region, activators can either directly interact with the general transcription factors. So this is the general transcription factor. They can either directly interact with the general transcription factors, or they can interact with the mediator proteins. And the mediator proteins then interact with the general transcription factors or RNA polymerase II to initiate the transcription. But in this case, I think there's someone else who wants to join. Okay, so the DNA loops back, and then the activators can interact either directly with the general transcription factors or with the mediator proteins, and then mediator proteins would interact with the RNA polymerase II or with the general transcription factors. And this is how transcription is initiated. But what repressors can do here, they can directly interact with the general transcription factors and block their expression. That's what we have already discussed. Just for refreshing your memories, I moved back to the previous slide. Okay, so this is the same mechanism, but that here, rather than the activator, it could bind with the general transcription factors. It's the distal control element contains the binding site for the repressor. So the DNA loops back, and repressor interacts with the general transcription factors and blocks them from activating the transcription or from initiating the transcription. So this is the third mechanism, how repressor proteins can operate. Now, these three mechanisms, what we have on the left side, they have nothing to do with the chromatin remodeling. They're not linked with chromatin remodeling, but we discussed how these repressor proteins can repress the or suppress the expression of the genes. Now, what we have on the right side are three different mechanisms, and these three mechanisms are involved in chromatin remodeling now. The first one is chromatin remodeling complexes. We have previously discussed this, that chromatin remodeling complexes have the ability to, these chromatin remodeling complexes have the ability to to change the they can either carry out sliding, they can carry out histone removal, they can carry out histone replacement. So they can do different types of works on this chromatin, and they can convert this chromatin from euchromatin into heterochromatin. So if this is converted into heterochromatin, this is the same mechanism as we previously discussed. When activator protein binds here, the activator protein recruits chromatin remodeling complexes which can carry out sliding of the nucleosomes or removal of the histone proteins. While when a suppressor or a repressor protein is bound here, this can do exactly the opposite of the activator protein. It would recruit such chromatin remodeling complexes which can lead to the compaction or condensation of the chromatin, and hence block the expression of the gene. The second thing they can do, they can recruit histone deacetylases. We have previously discussed that when activators bind here, they recruit histone acetylases, but when repressor proteins bind here, they can recruit histone deacetylases. And we know that the histone deacetylases, what they do, they remove the acetylation of the histone protein. And when acetylation is removed, we have previously discussed, the negative charge is gone, and the neutralization is gone, and now the DNA can interact with the tails of these histone proteins, and then it can get condensed. The sixth type of modification we have here is the modification carried out by the histone methyl transferases. So in this case, when a repressor protein binds to the chromatin or to the DNA, it recruits a histone methyl transferase. And we have previously discussed, we already know this methylation leads to the condensation of the DNA. So it converts euchromatin into heterochromatin, and this leads to the expression of the histone expression. So I think that's sufficient for today. If you have any questions, please.
Clear, sir.
Clear to everyone? If anyone else has any questions, please. We have 10 minutes left. सबको क्लियर है?
जी सर क्लियर है.
Okay. I think if no one else has any.
सर बाकी मुझे लगता है माइक्रोफोन लगा के इधर उधर हो जाते हैं.
मुझे भी ऐसे ही लगता है, बस बाकी I mean, सर को हाजिरी
सर समझ
आइए तो आप रिस्पॉन्स की नहीं. जुनैद कहाँ है? नहीं है ना जुनैद की तरफ से कोई रिस्पॉन्स नहीं आ रहा.
यकीनन गायब है.
जुनैद
जी सर आ गए.
अच्छा, रिफत और सुंदस.
सॉरी सर, एक्चुअली कॉल आ गई तो डिस्कनेक्ट हो गया था.
अच्छा, सही है. तो एनीवेज जी रिफत. रिफत नहीं है ना? रिफत ने ये काम शायद किया हुआ है.
रिफत नहीं है.
रिफत चली गई.
हाँ, रिफत चली गई. वो बस ऐसे हाजिरी लगाने के लिए आती है. ये उसकी मेहरबानी है, आ जाती है. आई एम रियली ग्रेटफुल टू हर ना. So anyways, I think thank you very much. I think nobody has any questions. समझ आ गया सब लोगों को clearly?
Yes sir.
समझ आ गई.
Okay, thank you very much. I will post the recorded lecture later then.
Yes sir.
Thank you.
Okay, thank you.
Allah Hafiz.
Allah Hafiz.